Good morning, everyone. Today we are here for the seminar of Vito Pastore. He has joined us recently at MaLGa as a postdoc. He obtained his PhD in bioengineering and robotics at UNICE, and he's been working for 2 years at a position for IBM Research Almaden in cellular engineering team aiming to use computer science and machine learning tools for making automated machines out of living cells. Today, he will be speaking to us about machine learning for environmental monitoring and cellular biosensing. Over to you, Vito.
Yeah, thank you, Gorby, for the introduction, and I want to thank the organizers for giving me a chance of describing my work. So today's talk is going to focus on machine learning application for cellular biosensing, and in particular for environmental monitoring. So as Gorby was saying, I've been working as a postdoc in IBM. So I just want to say a couple of words. IBM is the world's largest information technology research organization with more than 3,000 scientists and engineers. In particular, I have been working in Almaden, which is in the south of San Jose in California. And it's this beautiful building here on the right, which was first established in 1952. Let me give you a schematic overview of what we are going to see in today's presentation. I will start with an introduction, and I will provide a definition for this cellular engineering. Then I will talk about potential applications, focusing on environmental sensing. Later, I will introduce what is the problem that we want to solve regarding environmental sensing. I will describe a dataset and the microscope that we invented in IBM for the solution. I will also describe the features that we decided to engineer in an unsupervised learning pipeline, focusing on classification and anomaly detection. Finally, when all the machine learning tools will be described, I will give you an idea of how it's possible to use Plankton as biosensor, and we will draw some conclusions. Before I actually start with the talk, I just want to mention that the cellular engineering team, which I was part in IBM, is actually part of a bigger institution, which is the Center for Cellular Construction, which is a consortium of universities, including University of California, San Francisco State, Berkeley, and Stanford, and of course, IBM as well. So let me start with this. Let us consider a factory, a chemical engineering factory. We know this is basically a structure of production that takes some inputs and produce some useful outputs. Now, there are three properties which are very important for a factory, which are it can adjust size and the number of components. Of course, it will coordinate and collaborate with neighboring factories. And if more product is needed, it can grow and duplicate. Now, I want to make this rough analogy. A cell is not that different than a factory. And in fact, of course, it can grow and adjust its components. It will communicate and coordinate with the neighboring cells. Many mechanisms in the body are like that. And just can think about the scar mechanism where cells stop to grow when they are in contact. And finally, of course, the cell should and must grow and duplicate. However, there is a huge difference from a factory of a chemical engineering factory with a cell, which is in a factory, if I want to obtain a certain product, I know exactly what is the set of chemical processes and of structures. I don't know, I can think about tanks, which I have to apply in order to get my product. Instead, in a cell, this is not true. What I mean is, even if I know exactly the biochemistry of a cell, I don't know what should a cell look like in order to have a certain product. Ideally, what we would like to do is I want the cell to have a certain product. So I want a specific phenotype. It would be good if it would be great, actually, if I could be able to design the cell to have this product. So for example, to design the set of genetic perturbations to apply in order to obtain my cell, and to specifically understand how should the cell look like in the morphology, so to get my specific product. Now, cellular engineering aims exactly at this. In fact, cellular engineering is a new discipline which aims at transforming the field of cell biology into a quantitative discipline by adapting and exploiting tools from engineering, physical science, computer science, and of course, machine learning, so that we can design automated machines out of living cells. Now, in order to have this cell factory, we need two different points to be satisfied. We should be able to define individual cell structures, but also we should be able to understand that the cell is part of a bigger and more complex device, like a tissue or an organ in the organism. And so, about the definition of individual cell structure, ideally, like in a factory, you should be able to grow the components, the tanks and whatever with respect to the product. In a cell, we want to learn how to control the size of the organelles. Because if you think about that, like mitochondria are very important for the cell metabolism, and so like for the rate of production in the industry, in a factory. As well as we should be able to treat cells as circuits, and so we should be able to place them as chips, so to control cell interactions in a designed and specific way. So this is what basically is cellular engineering and what we are trying to do with the CCC. Now, there are different applications from industrial strain design to food protection, cell therapy, biofueling, and of course, we will focus today on environmental sensing. Every time that I give this kind of talk, people always ask me, why does IBM care for plankton? And every time I answer with another question, which is, why should you not care for plankton? Or why don't you care for plankton? And the reason is this one, because plankton is the basis of the marine food chain. So plankton means fish, and fish are primary animal source of protein for more than 1 billion of people. It has a huge economical impact. And phytoplankton also has produced 50 to 70% of our oxygen. So the answer is, if you like to breathe and you like sushi, which personally I do, then you should care for plankton. Now, why do I say this, that we should care for plankton? Because in the last 30 years, plankton population has decreased by 50% because of the increasing of temperature in the water. There are some techniques of analyzing and of controlling plankton's population, but are only global analysis tools. For instance, phytoplankton has some pigments like the chlorophyll, which can be seen as green. And so there are this global method of acquisition to monitor the density. But we actually want to do more. So we want to understand if the single plankton microorganism can be used as environmental monitor to actually tell us if there is a perturbation in the aquatic environment, so to be used as biosensor. And if this is possible, how would we actually implement such a biosensor? The idea is to establish a morphological and dynamic baseline. So we want to look at the plankton microorganism shape, at the way it swims in the water when it's healthy. And ideally, the feedback, the biosensor feedback comes from significant deviation from such a baseline, which should be indicative of perturbation, so that in the end, you can build smart environmental detectors to monitor the aquatic environment. Of course, when I say baseline, I mean that the shape and the way of swimming, so the behavior, we could say, of plankton microorganism will change according to perturbation. But it is intuitive that not all the plankton species will react in the same way. In fact, there are some species that react more and some species that react less. So some species that don't change their morphology. Of course, when you want to implement a biosensor, you are more interested in species that react more because you want to see as soon as possible the modification and so the presence of perturbation. Of course, this means that the very first thing you want to do is classification because each species will have its own baseline. As probably all of you already know, there are two main approaches when you do classification, which is a supervised one and unsupervised one. In a supervised approach, you annotate your dataset and basically you train a classifier in discovering the label of a testing data. And the disadvantage is that you need annotation, and of course, it can only recognize its training classes. However, it is very accurate if the dataset is large enough. Now, for our application, this is not really the best of the approaches because when we put our device in the water and there is a species which was not included into the dataset, we want still our architecture, our AI to work. So that's why we went with an unsupervised approach, which of course is powerful because it doesn't need annotation, so it's not expensive. And the training set can be dynamically extended. However, it's surely less accurate than supervised learning, especially when the species overlap because they have similar morphology. And it's strongly dependent on the features engineered. What I mean is that if you change the acquisition system, if you change the problem in this way, maybe you will need to work again on the features engineering. The pipeline that we propose for implementing plankton, for obtaining a biosensor from plankton is called Plankton Classifier, and it's composed of 5 different modules. We'll have a data acquisition module where basically you have the plankton samples. We acquire them with an in-house device that we invented, which is called lensless microscope, that produces plankton videos. These videos are used for detection and tracking, and the cropped image of plankton microorganisms are going through an image processor tool, which basically give us segmentation. And these segmented images are finally used for extracting some features, which are fed to an unsupervised partitioning, I mean, to a clustering algorithm. And finally, the label produced by the cluster are used to train an anomaly detector. And there will be a final pipeline, which I will show you in the end of the presentation for the actual implementation of the biosensor. So let's go with the dataset description. First of all, I want to focus on the lensless microscope that, as I was telling you, was invented by my team. Basically, this is just a very simple microscope with an LED, which is injecting light on a camera. This camera is like the one in your phone. We remove the glass and we constructed a well so that the plankton can directly float and swim on top of the camera. Of course, the shadow are projecting on the image sensor. And so you will see, what you will see, and I will show you now an example of video, it's the shadow projected by this LED. That's why we wanted to call it shadow microscope, but in the end we went with lensless because more importantly, since there is no lens, then there is no focal plane. And this means that you can see plankton swimming in 3D and in all the possible positions. They're not just a focal plane. Not only, this microscope is powered by a Raspberry Pi, so you can run several algorithms directly on board on the device. And this is very important for what we want to do. And it's also very cheap. You can build it with around $40. And this is an example of the video that you obtained. The organism that you are seeing here swimming is called Blepharisma, and it's a species of plankton that we took from California. Now, this is exactly the dataset that I personally acquired for this problem. So there are 10 different species. Some of them are more similar than others, and with a total of 5,000 training images and 1,400 testing images. I also wanted to consider a benchmark dataset, and in particular, we decided to go with the Woods Hole Oceanographic Institution, the WHI dataset, from which we extracted 20 species, and in particular 140 images per each species. Of course, this is not a color dataset, but it's very used in the literature, so we decided to use it for benchmarking our tools. Okay, so let me describe now the image processor. There are two different approaches that we applied regarding the fact that in some cases we have videos, like with the lensless microscope, but we don't have them for static images like the WHI dataset. In particular, when we have videos, we found that the most performing approach is just applying a background subtractor, which basically just means that each pixel, which is not changing its intensity for a certain number of frames, it's set as background and the rest is set as foreground. And then there are some image processing tools just to improve the results of segmentation after detection. And I just want to show you this video where you can see that actually the results are pretty accurate because this guy which is moving is a stentor and it's pretty much detected every time. You can even put a threshold on the area if you don't want the small guys, which are just not useful species, not interesting. And that is the image processor. If you have static images instead, there are two different approaches. You can decide to go with a classic image analysis. So like global bimodal thresholding, which for some of you maybe are familiar, it's like Otsu algorithm or triangle thresholding. Or you can go with deep learning. And for example, we tried with very high accuracy as well to train a unit. And again, the units, you can decide to pre-train some weights from a larger data set, like the ImageNet. You can either use the encoder that comes from another architecture, like ResNet or others. So it's pretty much powerful. But for our application, it was already enough, I have to say, an image analysis tool or the video that I showed you before. Now, let's give a look to the features that we decided to engineer so to feed the unsupervised, the clustering algorithm. So in order to fully describe our species of plankton, we decided to go with 5 different classes of features, which basically include shape features, which look at shape, but also texture to look at the way that the gray levels are distributed into our images. Now I will give you a very fast overview of these features. So in 1989, it was already shown that, of course, if you have species of plankton which are distinct in size and shape, then geometry is enough for the classification. And this is true, for example, for these two images that you see in the top of the slide. However, that's not true for the other two species of plankton, which are similar in geometry, though of course they have very distinct texture. So that's why you go with multiple kinds of features. And when I say geometric features, I mean perimeter, area, eccentricity, circularity, and so forth. Moment-spaced features are very, very popular in image analysis, and we decided to go with Who moments and Zernike moments. What I just want to mention is that these features are invariant with respect to rotation, scaling, and translation. And this is a property we really want because we don't want that any change in the position of plankton will change the features that we obtain, because we don't want to be dependent on the position of plankton in the chip, so in the water on top of the chip. Now, as I was telling you before, a texture is basically a description of the way that the gray levels are distributed into your image. And we decided to implement two different sets of features. One is the local binary pattern, and the other one is the hierarchic descriptors. The local binary pattern is an algorithm in which you apply a filter to your image, and then with a simple hard thresholding approach, you binarize the image, so each filter to obtain a binary number, a binary pattern. Then you put all of this binary pattern in a histogram, and the frequency of occurrence will correspond to your features. The hierarchic descriptors are as well very powerful in discriminating and for the classification of different textures. And they are extracted from grayscale co-occurrence matrix, which is a matrix in which the couple ij is basically the frequency of occurrence of the couple I and j for gray levels. So gray level I and gray level j. The Fourier descriptors are just a fast Fourier transform of your contour. In particular, if you want to gain some properties, you can decide to pre-filter your contour with another function. In this case, we use the shape signature, like the one you can see here on top of the slide, because we wanted to be invariant with respect to translation. Of course, the Fourier fast transform as it is invariant with respect to rotation. And if you divide for the first coefficient, you also get invariance with respect to scaling. It has been shown that the first 10 Fourier descriptors are enough to accurately describe images' shape. The last set of features is extracted directly from the histogram of gray levels, and is looking at very basic information like mean and standard deviation, but also the shape of the peak and the symmetry of the histogram. If you also have color information, then you should include them in this set of features. We have this for the lensless dataset, but we don't have them for the WHY, since the WHY is just in gray scale, it's not in color. So to summarize, we have a total of 131 features that of course came from a process of a larger number of features, and we found that these are the features that give you the best accuracy, so the highest accuracy for the dataset that we are studying. Now, finally, we can go with the results section, and we can look at the results of the clustering procedure. I will start with the lensless microscope results. First of all, another important tool is this one. If you really want to go unsupervised, you cannot tell the clustering algorithm how many clusters to use. So you have to find a way to infer this directly from your data. We decided to use different approaches, and one of the best performance was obtained with the partition entropy algorithm, where here is the equation, and this Uij is the degree of fuzziness. So basically, it's the probability that the sample I belongs to cluster J. If you look to the equation, you will realize that the lower is the partition entropy, the highest is the uncertainty in clustering. So basically, you want to take the peak of the curve that you obtain, changing the number of clusters as the most likely number of classes. And in order to ensure the convergence, or at least to increase the probability of convergence, we decided to use this procedure. We ran the algorithm 10 times, and then we take the mode of the number of clusters as the estimation. And here as a test, we selected some subset of our species, like 3, 5, 7, 8, and 10. And I have to say that each time the correct number of classes was found, so that the algorithm, at least for the lensless status, worked pretty well. So after you infer the number of classes, you can, of course, run your fuzzy k-mean, and this is the result. So in order to visualize the accuracy, I decided to take the principal component space. In the left, we see the annotation coming from the ground truth here. And on the right of the slide, we can see the results of the clustering procedure. The colors are assigned using the most overlapping class concept. So each cluster was assigned to the most overlapping ground truth class. And we obtained a purity around 93%, which means that 93% of points were put into the correct class, which is a pretty high accuracy for an unsupervised algorithm. Of course, this also means that our features really make our species much separated, also because the species themselves in this dataset are somewhere distinct in morphology, but there are some overlap. And I want to show you again this in 3D. So this is the same graph than before, but with a 3D exploration. And you can actually see that there is a very, very good separation with the features that we engineered. There is some overlap, but we will go on this overlap later on. So I put this slide, what about deep learning, just because I know that there is a tendency in literature. And actually, the reason I did this is because of this. So there is a tendency in literature to exploit deep learning. And so like to build expensive architecture to try what are the results with deep learning. But sometimes maybe this can overkill some of the results. In my case, in the dataset, in the lensless dataset, maybe already this approach which I showed you was enough, but we decided yet to go with a deep learning approach of features engineering. So what we did is we implemented a CNN, and in the CNN, we choose to gain a sort of invariance with respect to rotation by rotating our image 4 times, and so to multiple of 90 degrees basically. And then the features extraction part is run in parallel to these 4 images, and the features extracted are concatenated and going through the classification portion, where this layer here is just used for the deep feature extraction. So this is used for as features. And we repeated the same procedure than before. Of course, we have very higher accuracy, which is 99% with respect to 93%. But if the idea is to put our tools on the microscope, so on the Raspberry Pi, I'm not sure that the CNN can be run by the Raspberry Pi as well as the other just a fuzzy algorithm, I mean, just a fuzzy clustering. And so again, here you can see that the 2 images pretty much overlap. So ground truth on the left, the clustering results on the deep feature extracted on the right, they are almost overlapping, so are basically very similar. Okay, for the WHI dataset, again, I just want to show you that there is a purity of 89% for 20 species because the dataset is pretty much overlapping in several species. And again, if we want to look at the zoom in 3D for just 10 species, we can see that there is separation with our features. However, this time, many species really show a lot of morphology. And I mean, just to add something to the discussion, in the ocean, there are more than 4,000 species of plankton, and some of them actually are morphologically indistinguishable. And so you have to go. That's why in the beginning, I also spoke about dynamic, because sometimes it's not the shape that you can distinguish, but you can really differentiate with the way they move, so their behavior, let's say. Okay, now let's go back to the original point of my talk, which is about the biosensor and establishing the baseline for healthy plankton. With this slide, I want to give you a better insight of what I'm trying to say here. So on the left, if you look at this species of plankton, they appear different, but actually this is exactly the same species of plankton. It's called stentor. It's a huge cell. It's only 1 cell, which can be around 0.5 millimeter. And in fact, you can see it even with your own eyes in some way. And you can see that when it's healthy, so everything is going perfectly, it has this shape, this beam shape. However, when it's reacting to perturbation, it becomes sort of a ball. So what I mean when I say establishing a baseline is exactly this, because if I know what is the morphology of my plankton when everything is fine, and I see that it reacts to perturbation by changing its morphology, then I can actually use this in order to detect the perturbations. And here you can see a parallel coordinate plot, which is basically just 50 samples per each of the species. So each color is 1 species of plankton in the lensless dataset. And you can see, for instance, in this, which is relative to the paramount, that really there is a very good separation. So each species of plankton has its own baseline that can be used for later train an anomaly detection algorithm, but we will go through that. Again, the separation can be also appreciated by using an MGS curve, which is basically a sinusoidal fitting of your data. And again, different colors for different species, and we see that there is a pretty good separation between the species. Now, at this point of our pipeline, we have the clustering algorithm gave us some clusters and so some labels, and we can decide to use the labels provided by clusters to train a classifier and test on our testing set. And in particular, we decided to go with a very simple neural network since the data are separable almost. So we can go with a very shallow network with just a couple of layers of 40 neurons. And the testing accuracy is around 95%. Not only, we personally tested this on the Raspberry Pi, and it's compatible. It's very, very fast, even on the Raspberry Pi, so it can be run on top of our microscope. If you don't just, besides the accuracy, we can look here at the confusion matrix. We can see that it's almost diagonal. If you are wondering about this purple square here, this is the species Paramecium bursaria and Blepharisma. The reason is that Blepharisma is exactly looking like the Paramecium, but with a belly. And so since in our microscope species can go in 3D, as I was telling you, because there's no lens, there is a way that the Blepharisma turns where its belly is hidden, and so it's hidden, sorry, and so it's basically just completely undistinguishable from the Paramecium. However, they swim very differently, so one could use behavior for this separation. Again, the rock curves is a very similar concept. So the receiver operating characteristic curve, when you have random guess, will be the bisector. Instead, when you have perfect guessing, it will be this square, this unit square. And most of our classification are close to the unit square. Okay, now we know that there is a baseline of healthy plankton that we established. And what we want to do is basically detect every deviation, significant deviation from the baseline as an anomaly. As an anomaly, there will be probably, and we will see how, related to environmental threat or perturbation. But now we need an anomaly detection algorithm. We decided to implement a particular neural network, which we call delta-enhanced class detector, where basically we have the original sample, and we decided, now this is a binary algorithm. So each species will have its own DEC detector, of course, as every anomaly detector. And now from the species that we are training, we extract a subset of samples whose number is a parameter to hypertune for your application. In our case, 25 was a good fit, so it gives us good performance. And the idea is you feed the sample, but you also feed the difference with respect to the sample and the subset in the feature space. So there are some layers of independency. In the end, they are concatenated together. And the idea is that you leave the neural network decide if you want to give more weight to the sample or to the distance in the feature space of the sample from some representative samples that are randomly extracted from the training species. And actually, the DEC detectors works pretty well on our dataset. We obtained an overall accuracy of 99%. When I say overall, I mean classification, because of course you can use an anomaly detector to detect in class, and also anomaly detection. For testing anomaly detection, we consider it for each species that was trained in the DEC detector, the remaining 9 species as anomaly, because of course they are in the concept that in the way we are describing the problem. We also decided to use a state-of-the-art anomaly detector as a comparison. And so we used the one-class SVM. In particular, we obtained still a high accuracy, around 93%. However, while with the DEC, the minimum that you obtain is around 92%. In the one-class SVM is around 78%. And it happens again in the species Paramecium and Blepharisma. We also try to understand what was the robustness of the DEC, which means now the species are pretty much separated, right? But what happens if a species is quite similar to one of our training ones? So the idea is let's build a synthetic and in silico species, which is basically just a linear combination in the feature space of of the 10 species that we possess. Now we will increase, if the DEC detectors is being trained with species X, we increase the coefficient of X by 10%, from 10 to 90%. And we can see that up to 30%, basically the similarity is just 98%, so there is no real change. But After 50%, we start with a decrease in the accuracy, which is significant, and we reach a minimum of 37% when there are 90% of similarities. Of course, still 37% is not completely bad because we want to detect a significant amount, so a non-zero, let's say significant amount of anomalies, but really 90% of similarity, I mean, these are results expected. Now, all the machine learning tools have been described, and we can focus on how you can implement a biosensor from Plankton. The idea is this one. So I talked about the DC detectors. The idea is you can put these anomaly detectors in parallel, and the idea is if all of these detectors reject a sample as anomaly, then the sample will actually be recognized as a global anomaly. If only one of these DC detectors responds, this sample will belong to the species that is correspondent to the DC. However, if more DC detectors are activated, so recognizes in class our sample, then this sample is interesting because it shares in some way, it overlaps with the species that we are using in training. And so it should be used for retraining. With this slide, I just want to highlight that you don't really need to use the DC detectors. I mean, that gives us the best performance, but in general, you can use whatever anomaly detector you want, but just put them in parallel and define a global anomaly only when all of them reject the sample as anomalous. Okay, finally, this is the final pipeline, which I was telling you about in the introduction. So everything I told you by right now is up to the Plankton classifier, which gives us these DC detectors or anomaly detectors in general, which we will call smart detectors. So now, when a test sample comes into our pipeline, it can be recognized as in class, so you're just using for the update, or it can be recognized as a global anomaly. A global anomaly, so a percentage of the global anomaly will be every day of acquisition stored and sent to an expert, which will have a graphical user interface software for visualizing these images and deciding if they are, because there are two main possibilities. An anomaly can be just an unseen species, so a species which was not included into our dataset. Of course, it's anomalous with respect to the dataset. Or it can be one of our species whose morphology changed so much because of perturbation that is no more recognizable as belonging to the species. And this is the case of the stentor, which I showed you before, that was becoming a ball. And so if it's actually an unseen organism, you can collect and store this sample. And in the end, you can perform again the procedure to obtain on these new samples some new detectors and start again. Or if there is there is a frequency of actual anomalies, so actually changing the morphology for perturbation, you can raise a flag of potential environmental perturbation. Now, in order to actually prove that the Plankton classifier and the pipeline, which I just described, can reveal unseen species, we decided to perform a leave one out test. So we excluded one species for all of the species, and we used the remaining 9 DC detectors in the way which I just described. And every time you can obtain a detection of the 10th species as unseen, so as novel, with very, very high accuracy. There is a minimum in the case of paramecium, which is species number 10, where the result is 70%, but just because many times the blepharisma detector was responding to the paramecium sample. However, 70% is still pretty much enough so that you can train a new DC detectors and then coming back to the original accuracy, which I showed you in previous slides. Okay, so I can conclude. And what I show you in this presentation is that machine learning allows for excellent classification of plankton with both supervised and unsupervised approaches. About the unsupervised approach that we choose because of the training, because we don't want to be dependent on the training samples and we don't want to annotate, we selected a list of 121 features, which allow for very good separation for the investigative species of plankton acquired with a lensless microscope. Our method can be used to establish a baseline for healthy plankton, and an anomaly detector approach can be used for revealing significant deviation from such a baseline, which will correspond to perturbation. So we can build in this way, our microscope powered with this algorithm, the Raspberry Pi can be put in different places in the world. Maybe we can also define an IoT structure so they can communicate with central servers and they can exchange information so you can obtain smart detectors to monitor the aquatic environment. As a real final couple of slides, I just want to mention that all the described tools for the unsupervised learning of Plankton have been actually used in other applications, in particular during my postdoc. So the image segmentation tool, I also been working on, for example, philopodia detection, so segmentation of cell, where philopodia is quite interesting because these are these filaments which are applied in the motility and are also involved in cancer of cell. Also, as you remember in the beginning, I was telling you that cellular engineering in a certain way aims in understanding what is the set of genetic perturbations to apply in order to obtain a specific phenotype, like having a product. And now it is very important to obtain a mapping of genetics into morphology. And on the right of the slide, we can see an application of this, where we actually found a way so we actually applied all of these tools to data-driven modeling from cancer cells. And finally, synthetic engineering is another very interesting application of cellular engineering. Here in particular, we were studying yeasts. And when you engineer an yeast to have a product, this product can be stored into the vacuoles. So it's very important to understand how genetic perturbations can change the structure of our vacuoles, because they can affect the productivity of your industry or for your industry in having this product. Okay, so I just want to finish acknowledging my group, Sujoy, which was my colleague, my postdoc, who took care of the dynamic explanation and study of plankton. Simone Bianco, who is the PI, and Thomas Zimmerman, who is the inventor of the microscope. And thank you all, and I welcome every question.
Thank you, Vito, for the very interesting seminar. Opening the platform for questions. Firstly, I would ask those of you who are on the YouTube part to post the questions as comments. Meanwhile, everybody on Zoom, feel free to ask your questions.
I have a couple of curiosities. Thank you, Vito. It was I already knew something, but it was very interesting to hear the whole story. The first curiosity is about this no lens sensor, and you were saying that it's special because it does not. I'm not sure I understood the fact that it doesn't have a focal plane. So you're meaning that you can see the 3D evolution of the cells.
Yeah, because we removed the lens, so there is actually no lens, and it's just like you only collect these shadows of plankton. So basically, you can obtain even if the image is 2D, we also had a version 3D of our microscope. You don't have a plane; you can see them moving in 3D, and this is from a certain point of view is an advantage, but from the other point of view, this caused something like the stuff I was telling you when the blepharisma with the bell is not distinguishable from the paramecium. Because since they can go into 3D as well, they have this drop available, and you see all the drop. Sometimes, they can be farther or closer to your focal, to your image sensor, and so this can give some problems. But it's good because you have dynamic.
Yeah, to be sure I'm understanding, of course, this is something that you're saying in comparison, for instance, with traditional microscopes, right?
Yes,
where everything happens bidimensionally.
Yes.
Okay.
Yes.
Okay. And partly related with this, maybe you already mentioned some answer to my question, but I just wanted to be sure I understood. Why did you? Is there any specific reason for choosing the analysis at an image level and not considering image sequences, where in fact those issues related with occlusions or some weird movements could be detected?
Actually, it's a very good question, and I think this was just this is a preliminary, let's say, analysis where we just wanted to look at morphology in static, because if you use our microscope and you get videos, I agree that the best way is actually to look at the image in time and not really extract features anymore, but just looking at this sequence. However, we wanted to be sure to have a method that can be applied also to static image to benchmark with literature already, so to have kind of a benchmark. Right. But I think that the step 2 will be exactly the one you are proposing, which I don't think I will take care of that anymore. But I think that they will actually do that. Yeah. that will go.
Yeah, in a sense, I have in mind some kind of extension of what you already did. Same features, maybe tracked over time, maybe in some small temporal steps.
We did
nothing
that as
major,
well.
but
Yeah. We did also that. We did some tracking, and also, just because, as I was telling you, since morphology is never enough, because there are so many species of plankton that many of them will be undistinguishable. So, and some of them don't necessarily need change morphology when are exposed to chemicals. They only change the behavior because, for example, just to say they die, for example, and you can see this because they stop moving. So the idea of tracking features, dynamic features, we have been working on something like that already. And in fact, it's very accurate. And it solves some of the problem that we were having with morphology.
Okay, thank you. Thank you very much.
Thank you.
So are there any other questions? Okay, I don't see any questions also on the YouTube. So I would like to thank Vito for a very interesting seminar, and thank you.
Yeah, thank you for inviting me. Thank you.